The overall goal of this method is to monitor the entire sexual reproduction process in fission yeast by light microscopy.
This method describes the preparation of fission yeast cells for time-lapse imaging of various stages of the sexual life cycle, such as mating partner choice, polarized growth, cell-cell fusion, meiosis, and sporulation. The main advantage of this technique is that it provides single-cell visualization and so overcomes the problem that cells do not enter sexual differentiation synchronously.
This method is very simple, but visual demonstration is useful to show how to mount several strains in parallel for microscopy.
Demonstrating the procedure with my PhD student Omaya Dudin will be Laura Merlini, a postdoc in my lab.
There are several variations to this protocol which can be tailored to study various stages of the sexual life cycle, using either homothallic or heterothallic strains. In this demonstration, homothallic H90 strains will be used. In these strains, mating type switching takes place during the last mitotic division after nitrogen deprivation, resulting in a mix of the two mating types. In the evening, 2 days prior to the mating experiment, inoculate freshly streaked strains from solid media into culture tubes containing 3 milliliters of minimum sporulation medium with nitrogen, or MSL plus N. Incubate overnight with shaking at 25 degrees Celsius. On the following morning, if necessary, dilute the cell suspensions.